This conference will now be recorded.
First Wednesday, we have our third exam. We'll cover everything up through what we finished today. Where we had left off, if I can get the projector to work here. Where we had left off, we'd been beginning to kind of talk about moving gases around in the blood. All right. And one of the things that we'd mentioned a few times, of course, is that oxygen and carbon dioxide necessarily have to travel differently because oxygen will not really diffuse in water, but really any liquid, whereas carbon dioxide does so readily and quite well. And so they will be moved around in different forms. For carbon dioxide, we move it around in not in the form of CO2, but in form of either bicarbonate or carbonic acid. And the means by which that happens, right, is called the chloride shift. So let's take a look at a cartoon here. We'll explain what's going on with this. So for right now, we're only worried about what's happening here with the blue arrows. Okay, the red arrows really aren't super important for us at this moment. So we're thinking about taking carbon dioxide from the extracellular fluid and offloading it into the blood. So some of that, if I can find my cursor, some of that does just end up as dissolved CO2. Okay, but the vast, vast, vast majority of it, right, travels as bicarbonate or carbonic acid. Okay, as part of this buffer system in the blood. Okay, and there are two means by which we can convert carbon dioxide in the presence of water to bicarbonate or carbonic acid. We have the slow pathway, which occurs outside of the red blood cell, or, and this is where most of it travels, we have the fast pathway, which occurs inside the red blood cell. The reason we process here is that we balance the offloading of this bicarbonate. So the bicarbonate leaves the red blood cell and enters the plasma. We balance that negative charge leaving the red blood cell by picking up a negative chloride charge. So the charge is balanced, right? And chloride shifts from being outside the red blood cell to being inside the red blood cell. That's all that comes down to for us. We do also transport some, you may remember we talked about carbaminohemoglobin, where hemoglobin will, if it has really no other option, will pick up carbon dioxide, but it doesn't like to. Okay, but most of it, the vast majority, is going to be transported around out here in the plasma, predominantly as part of this carbonate bicarbonate buffer system. And this is all well and good in the systemic capillaries, right? Where we're out in the body, we're dropping off oxygen, but we're picking up carbon dioxide. In pulmonary capillaries, it's a little bit different, right? Because here, our job is not to pick up carbon dioxide, but to drop it off. And so all we're going to do is those processes in reverse. Okay, so just like before, and again, I'm not so worried about the red arrow at this moment. As before, right, we have to now either offload this dissolved carbon dioxide, which will just diffuse out of the blood, right? That's diffused in, it diffuses out, everybody's happy. Or, right, we take this carbonate or carbonic acid bicarbonate buffer, right, this sort of back and forth. We have to turn that back into water and carbon dioxide. Okay, and just as there was a slow method to do so outside the red blood cell and a fast method to do so inside, here going the opposite direction, we have a slow method outside and a fast one in. Okay, and literally all we do is just go, we just take these arrows and we turn them in the opposite direction. That's it. Okay, we once again experience a chloride shift. Okay, but now it's going in the opposite direction. Right, the red blood cell is picking up bicarbonate and dropping off chloride. Again, just so the charge is balanced. Right. So really, if you kind of just imagine that because the transport direction is the opposite, that all you do is take the little arrows in your chemical equations and turn them the other direction. That's really all there is to it. Now, there is one additional little caveat here, okay? And this is what we call the Haldane effect. And the Haldane effect is not specific to this particular relationship. It has to do with the partial pressures of various gases and the receptibility of fluids to others. Here in this particular case, the amount of carbon dioxide that your blood will carry is affected by the partial pressure of oxygen. Okay. So if there's lower partial pressure of oxygen, right, your blood will pick up more carbon dioxide. Right. The reason we bring that up is that this is a handy dandy little thing if we're trying to pick up carbon dioxide in places where we're dropping off oxygen. Right. If we take oxygen and remove it from the blood, right, we lower that partial pressure. We make the blood more likely to pick up carbon dioxide. That's our friend Haldane, right? Now, there's a kind of a knock-on effect here, right? This is the Bohr effect. And the Bohr effect is basically that as the blood picks up more carbon dioxide, it's more likely to drop off more oxygen. So again, we have this kind of feedback system where if we're in a place where we have a lot of activity, a lot of cellular activity, we're going to generate a lot of carbon dioxide, right? And we're going to need a lot of oxygen. And so this is just one of the body's ways of kind of ensuring that oxygen gets to where it needs to go, right? And doesn't get dropped off in places maybe where they don't need extra. So something we talked about in the circulatory system a little bit, and we talked about it a little bit in ANP1, was this bicarbonate carbonic acid buffer, right? And the idea here, again, just to simply maintain homeostasis with regard to blood pH, right? We talked about the idea that if this blood pH gets much farther away from 7.4 than like 7.35 or 7.45, bad things start to happen in a hurry, okay? And so what we'll do to resist that then is that as we get more acidic, right, we end up forming more carbonic acid. As the blood gets more basic, we end up forming more bicarbonate, right? Just by exchanging these hydrogen ions with either bicarbonate or carbonic acid. And in that way then, right, because the transport of carbon dioxide in the blood as bicarbonate carbonic acid is so vital to the maintenance of pH, this helps explain a little bit why the partial pressure of carbon dioxide in the blood is always fairly high, right? It's fairly consistent. It's about 45 to 40 millimeters mercury, no matter where you are in the body, right? No matter what point of circulation you're in, it's always roughly around that same concentration. And the reason here is because we can't get rid of it, right? If we got rid of it, we wouldn't be able to sort of balance our own pH in our blood, and we would go south in a pretty big hurry. So while it is important to offload carbon dioxide, and the breath that you breathe out does contain a lot of carbon dioxide, a lot of it does remain in your blood, right? A lot of it does not actually ever go anywhere. Now, what we do to sort of keep an eye on oxygen needs, oxygen delivery capacity, all sorts of things, we monitor the blood for a handful of different factors, different chemical kinds of compositions in the plasma. We look at oxygen content, and we look at carbon dioxide content, but really more than anything else, we look at pH. And the reason that your brain, your hypothalamus, looks at pH more than anything else is that pH is intrinsically linked to PCO2, right? The more CO2 you have dissolved in your blood, the more bicarbonate, carbonic acid you would end up introducing to the blood. Typically then the lower the pH is going to be associated with that, okay? So adding carbon dioxide to your blood lowers the pH both globally and locally. So, right, when we think about how rest rate might affect this or vice versa. Okay, slow, shallow breathing typically leads to a rise in carbon dioxide, right, and a drop in pH. Rapid shallow breathing, rapid deep breathing rather, leads typically to decreased carbon dioxide content in the blood and a more alkaline blood. And so one thing that you can do, and your body does do, to try to balance this out a little bit if the buffer system isn't adequate at the moment, is change rates of ventilation. Okay, if your blood is getting a little acidic, you end up taking deeper, more rapid breaths, and vice versa if your blood is becoming too alkaline. As far as where this actually happens, like most things, we have a couple of different control centers. There is some input from higher brain centers. Obviously, we talk about being able to produce speech, right, as a voluntary action, which has to do with the expelling of breath. You can also consciously take deep breaths or shallow breaths. You can do all kinds of things with those higher brain centers. But most of the sort of the workaday, day-to-day, in and out of ventilation is from the brainstem and from the hypothalamus. Okay, chemoreceptors within those two groups. So as far as the sort of the brain kind of neural controls, we really think about the medulla and the pons. Okay, in particular the reticular formation, which you may remember we talked about is amongst other things being kind of a spam filter for things coming up, but also a regulatory center for repeated or stereotypic actions coming down. Okay, but also two different respiratory groups, specifically in the medulla, the VRG and the DRG, or the ventral and dorsal respiratory groups. VRG is going to set what we call eupnea. U here, EU meaning true, nea meaning breath. So eupnea is your resting breathing rate. Okay, and this is somewhere around 15 breaths per minute, maybe 12. Okay, and this is just, when you're just sitting around, you're not really not really doing much of your sort of standard respiratory rate. And again, this is pretty consistent from person to person. The average breath takes about 5 seconds. So we're sitting somewhere around 12 to 15 breaths per minute. The DRG is a little bit different. The DRG is going to be the dorsal respiratory group, is going to kind of modify the activity of the VRG. So we're going to be getting information from chemoreceptors, right? So we're going to be looking at like pH of your blood, for example, or PCO2 or PO2, primarily pH. We're going to be looking at stretch receptors from the lungs themselves, okay? And all of that information kind of gets shuffled amongst other places to this DRG, and the DRG says, okay, well, we got this problem or that problem. We need to modify our respiratory rate to accommodate that, all right? So again, we kind of go back to some of our examples before. Blood becoming too alkaline, the DRG will tell the VRG, okay, let's do a little more shallow here. Okay, let's get a little more CO2 in the blood to slow it down a little bit, and we'll drop that pH back to where we need to go, or vice versa. So if we kind of zoom in here on our brainstem, right, here's the, there's pontine receptors here, we'll talk about this in a second, but down here in the medulla, we have the DRG and the VRG. And the VRG, right, is going out directly to your respiratory muscles, but the DRG kind of informs the activity of the VRG. By that same token, right, if you look at the arrows here from the pontine group, which is the next of our respiratory centers, it's going out and it's communicating with both the VRG and DRG. Okay, and again, much like the DRG here, just kind of modulating or modifying or slightly adjusting the activity of that VRG. And much like much like the reticular system in general, right? It also kind of plays a role in smoothing transitions between contractions and relaxations, right? We talk about that reticular system kind of preventing jerky movements, right? And sort of damage to this particular region of the pond, the kind of leading to sort of shaky or jerky movements, kind of doing the same thing here as well, right? We're kind of easing into and easing out of inspiration and expiration. This is also where we kind of, again, think about where we're at, right? Coming from the higher brain centers down through the pons here. This is also where we're going to be doing a lot of that modification based on conscious behavior. So breath control for speech or other vocalizations or what have you. Now, how the VRG actually sets eupnea is not 100% understood. We're pretty sure we know exactly how this works, but again, it's one of those things that you don't want to say 100% because you never really know. The sort of most common and almost certainly the correct answer is actually pretty simple. It's basically we have a network of two different pacemakers, one that are set kind of in a little circular loop, such that one inhibits the other, right? And then when the one is no longer being inhibited, it inhibits the first one, and then it goes back and inhibits the next one, and kind of in this little sort of relay loop of kind of turning the other one off. And that's pretty typical of how most central pattern generator type structures work within the brain. This is almost certainly a CPG. Sometimes they're multi-neuron, usually they're pairs of neurons like this one, but that's almost certainly what this ends up happening. And this is in the medulla. Now, as far as how do we influence rate or depth of breathing, again, we've kind of touched on some of this already, right? Some of the factors that come into this. The rate here is largely determined by how long do we kind of put our finger on the button to breathe in, right? That's what's really going to set our base rhythm. It's not so much, again, if you remember kind of the idea that inspiration is always an active process, expiration is often passive, right? This might kind of follow directly from that, right? As long as you've got the button on the inflate, our finger on the inflate button, that's going to be breathing in, and we'll set everything else based on that pace. We're going to alter that, of course, based on PO2, PCO2, and pH, right? PCO2 here, in terms of our sort of chemoreceptors, we spend a lot of time thinking about PCO2. PO2 really is, I mean, we monitor it, right, honestly, but it's not the biggest factor. It's kind of down on the list. And really, honestly, it doesn't really come into play until things are really bad, right? PCO2, on the other hand, we keep a really tight monitor on, right? Because PCO2 is really is simply a better canary in the coal mine for us, right? Because again, remember, we've got this venous reserve of oxygen, right? We only ever usually drop off 25% of the oxygen we're carrying. So if PO2 wiggles a little bit, right, goes down a little bit, we not really in trouble. Okay. PCO2, right, is much more a factor of what are we using, are we in trouble kinds of things. So we spend a little more time with that. If we get hypercapnia, which is elevated PCO2 or serum CO2 levels, that's bad, I guess is the best way to put it. Typically what's going to end up happening there is we get in the central nervous system, in the brain in particular, but in the central nervous system in general, CO2 tends to accumulate and mix with water. And CO2 and water makes carbonic acid. And carbonic acid dissociates, right, makes bicarbonate and free hydrogen ions, and you acidify your brain. Okay, but, right, this is not such an immediately bad thing because you've got in your in the brainstem itself, in some of those chemoreceptor regions, we're monitoring for this dissociated hydrogen, this basically drop in pH. Okay, and that's going to be what influences sort of a uptick in depth and rate of breathing, trying to lower that PCO2. Right. We lower that lower that PCO2, right? We kind of shift our carbonic acid back the other direction. We reassociate it. We make more carbonic acid, right? We make less bicarbonate, make more carbonic acid. And in doing so, we lower the pH of the blood, or raise the pH of the blood, rather. And again, if you're trying to kind of follow along, this diagram might help. It's got a little bit more in there than we worry about, but it is a pretty decent summary. Hyperventilation is, well, you've probably almost certainly seen someone hyperventilate. Maybe you've hyperventilated yourself. The issue with hyperventilation is that we have, because you breathe so deeply and so rapidly, you basically offload all of your carbon dioxide. Right? You become fully oxygenated, which is great, right? But you offload so much carbon dioxide, you end up actually hypocapnic, right? We have hypercapnia with too much, hypocapnia is not enough. And with decreased blood CO2 levels, this hypocapnia, what we get here is typically widespread vasoconstriction. Okay, constriction leads to dizziness, fainting. If you fall and hit your head, you can die from this, right? So one of the reasons that you give somebody that's hyperventilating a paper bag to breathe in is you're trying to get them to rebreathe that air, right? Again, the danger there is that they're breathing, and pumping all that carbon dioxide out, okay, and they're not getting it back in. If you restrict them to breathing in the air they just breathed back out, they're going to bring all that carbon dioxide back in, and they're not going to get to that dangerous hypocapnic level where they're going to see cerebral ischemias, fainting, that kind of stuff. Okay, so it's not one of those things that you would see on TV and think, oh, that's never what you would really do. No, that's literally what you're supposed to do. Apnea is a term you've probably heard primarily in terms of sleep apnea. Apnea is just when you're not breathing, right? So you put a in front of a word, it means not. Apnea, right? Breathing. So apnea here is not breathing. If you have excessively low PCO2, you will temporarily stop breathing, because again, you have no need to continue to breathe if you have not any carbon dioxide to offload. So it's not necessarily great. PO2, right? Again, partial pressure of oxygen. We do monitor it. It's not the most important thing, right? We don't see catastrophic bodily changes deriving from low CO or from low PO2. The only thing that would ever really come out of this is if you're extraordinarily low, right? Down below, say, 60 millimeters mercury in terms of PO2, right? Again, remember, when we leave the lungs, we're at 100, right? So to get to 60, we got to be pretty low. And even then, right, it's only to simply say, okay, well, we're going to breathe. We're going to breathe a little bit more, right? We're going to breathe heavily. We're going to breathe deeply and breathe a little more rapidly to try to get some more oxygen in there. It's really not something that comes into play unless you're, say, exercising or something like that. Right? Again, our primary factor there is monitoring PCO2 rather than PO2. And in terms of where our sensors are for PO2, primarily here in the carotid arteries, in the sort of the main body of the carotid, so the branching point of the common carotids, called the carotid bodies. This is where our most of that PO2 sensation comes from. And then, of course, arterial pH, right? Primarily here, pH is a factor of carbon dioxide dissolution in the blood. So it's hard to separate pH from carbon dioxide in that way. It's rare that you would find the two not linked, I guess. It's rising PCO2 and declining pH or vice versa. Almost invariably the same thing. And so we would treat, of course, declining pH in the same way that we would treat elevated PCO2, right? We're going to sort of slow and shallow that breath a little bit. Or rather, the opposite, sorry. Okay, so summarize the whole thing. Most important thing we're worried about here, CO2 levels. We don't really honestly worry too much about PO2. We worry about pH, but really pH so tightly associated with CO2 that they're essentially the same thing. All right. The only time that we really see any change associated with PO2 is when it gets extraordinarily low, right? We're starting to dip into that venous reserve a little bit. At that point, we'll begin to up respiratory depth to try to get more oxygen in the blood. But that's a pretty far fall from sort of resting PO2. It's hard to get there without really working at it. But if we get there, we'll get there. Remember, we go about 60%, maybe a little less, okay, of our starting PO2 for us to get there. You do obviously have control here above the brainstem level. We've talked about this a little bit, right? Hypothalamus plays a big role in this. And if you remember from our discussions about the hypothalamus back in ANP1, right, the hypothalamus and the brainstem, there's sort of at first blush, right, a lot of overlap between the two, all right? The reason being the hypothalamus is kind of the foreman, right, who decides what needs to happen. He gets all the information, or she gets all the information, right, and decides who needs to do what job based on that, right? And the brainstem, right, the pons, the pontine centers, the VRG, the DRG in this case, these are very much the workers, right, that are carrying out the orders of the hypothalamus. So again, it's hard to separate the two, right, when one is very much kind of in charge or overseeing the others. But the hypothalamus, because of its kind of interesting position within the brain and its relationship to, say, the limbic system or some of the other things that are in there, it also gets a lot of directive, might be the wrong word, but input from some of these higher brain systems. And so we get, say, a gasping behavior or something like that associated with a surprise or a shock. It's an emotional state kind of feature, but derives from input to the hypothalamus. Obviously, of course, you can decide to, say, hold your breath, right? You can decide to take a deep breath. You can decide to sigh, I'm doing that a lot these days, right? So there is some cortical control, I guess we can apply to that as well. Again, in interrupting eupnea, okay, altering eupnea, things like that, but not really capable of overriding it. Let's put it that way. You can't hold your breath until you die, right? Might have tried as a child, okay? You'll teach mom and dad a lesson, right? But you can't do it. There are a couple of useful reflexes here to think about. The irritant reflexes are what give rise to coughs and sneezes. Anything that is going to be an irritation to the mucosa of the upper part of your airway will give us this irritant reflex. And the only thing that distinguishes between whether you cough or sneeze is location. Okay, the exact same stimulant will cause a sneeze in the uppermost airway and a cough in the lower parts of the upper airway. And again, the idea there is simply you've got something that's triggering some irritation. You're trying to force it out, right? That's really the only thing that's going on there. So this is going to require not only sort of a forceful exhalation, right, but also remember it's that going to contract that trachealis muscle. Okay, going to narrow the airway, try to make as much velocity out of that airway as possible to try to force whatever this thing happens to be out. The other big reflex here for us is Herring-Brier. Herring-Brier, sometimes called the inflation reflex. This is going to be something that is in relation to stretch for the lung itself. Basically, the idea behind Herring-Brier is that you've got a safe in all of your bodily tissues, right? In all of the physiology that you've got, there's always we'll call a safety factor built in. Basically, the idea is, yes, you are strong enough to rip your muscle off your bone, but you're physiologically incapable of doing it because of a safety factor, right? Here, our example of a safety factor is Herring-Brier. And the idea behind Herring-Brier is that as your airways expand and expand and expand and expand, of course, they're going to get to a point where you might become in danger of rupture. Herring-Brier gets us within that safety factor of doing that and forces us to exhale. Okay, it's just basically there to prevent you from rupture alveoli or in the parts of your airway through overuse. It's not really something that you will encounter in your day-to-day life. It's a protective mechanism. It's not a regulatory one, I guess, in that regard. So if we kind of put all this together then, right, we've got Herring-Brier here, right, our stretch receptors in our lungs, which will inhibit the respiratory centers, the pontine, the VRG, the DRG, okay? You've got your irritant receptors here, right, again, for your cough or your sneeze, again, doing the same basic thing, right? Inhibiting this, causing you to exhale, right, or to stop inhaling, if nothing else. You've got chemoreceptors, right, in the aorta, here in the carotid body, okay, up here in the hypothalamus, okay. In this particular case, right, declining O2, but primarily or a drop in pH, using a stimulation there to breathe in more, right? Yeah. Or things like your higher brain centers here, right, where you override some of this basic eupnea, right, to hold your breath, or to breathe in, or to breathe out, or to yell, or to speak, or to do whatever you're going to be doing, right? Lots of different little places with lots of different little inputs, but all coming to the same place. You will notice if you ever travel somewhere that's reasonably high above sea level that it is, in the short term, difficult to breathe. I guess we'll put it that way. And the reason being that above a certain altitude, the concentration of oxygen in the air is significantly lower than it is at sea level. And we're built, as organisms, we're built to operate more or less at sea level. And if you're to go significantly higher above that, you can actually get altitude sickness, or something that's called mountain sickness, which is going to be basically the same thing that happens to you if you're suffocating. You get headaches, you get dizzy, you get nauseous. Again, remember, your brain takes everything that doesn't seem right and says, well, you probably eat something bad, let's throw up. It's kind of its go-to solution for everything. And it can, in fact, kill you. Right? Now, being at significantly low partial pressures of oxygen in the atmosphere will kill you. You can acclimatize to it, though. We've talked a little bit about acclimatization in terms of circulatory system, right? With increased rates of hematopoiesis, right? We're making more red blood cells. In terms of your respiratory acclimatization here, a lot of it has to do with adjustments made by your chemoreceptors. They become more receptive to changes in CO2, for example. Again, just kind of starting the transition to deeper, more rapid breaths earlier, I guess, is what that's happening. Okay. sort of not allowing kind of the partial pressures to change more than they would at sea level, we're going to just kind of begin adjusting earlier on. And it will eventually get to a point, you'll be about 2 or 3 days where you're there before you don't notice your change to eupnea. You will have changed eupnea, but it will take you a while to kind of get there. One of the issues with that, of course, is that no matter what you do, you will still always have lower than normal oxygen saturation. You just can't get around that. You can make all the red blood cells you want to, you're still going to have lower than normal oxygen saturation. Functionally, what that means for you is simply you have less venous reserve. Okay, so you won't notice it when you're walking around, when you're sitting in the classroom, when you're doing your normal day-to-day things, but you'll notice it like if you go for a run, right? If you can run 10 miles at sea level, you're probably going to run 7 or 8 at altitude, right? You're just not going to have that ability to kind of expand beyond that resting activity anymore. All right, so that's all I got for you. So on Wednesday, of course, we'll have an exam. So same deal as always, right? It'll be online, 50 minutes, same kinds of questions, all that kind of good stuff. Study guide got posted last week, so make sure you take a look at that. Other than that, I got nothing for you except to say I'll see you on Thursday.